I know. It's early in the morning. These 24-hour operations can be a little challenging to get adjusted to. But we have to take advantage of the limited time we have out here. So the ship is, I mean, when you're in this control room here, you don't even know whether it's day or night or where you are. You can be anywhere on the planet. It's almost like aside from the feeds we have coming in, it's almost like a sensory deprivation chamber in here. It's pitch black and aside from the screens and such, but
Yeah.
You mean we're not on a spaceship and we're heading to Mars?
we
I
might
thought
be
we
on the
were.
spaceship, Chris.
Oh.
I mean, I don't have any way to prove or disprove that. I love when people try to describe what we're going to do when we're out here, and they all are like, oh yeah, take plenty of pictures, and
Oh yeah.
you know, and you're gonna get in the water. And I was like, well, we don't get in the water at all, and there's about a bazillion pictures coming in over the feeds that anybody can see anywhere in the world, so there's not really any point in taking any pictures.
Yeah.
And they just give me a quizzical look of, I don't get it, I don't understand.
I'm with you, Andy. I said I'm not gonna take pictures. Maybe I'll take 1 or 2 pictures of the sunset with my old phone, but the best pictures are gonna come from
the live
Hercules, yeah. But for those audiences out there, listeners, viewers, we only have about 30 minutes for our ROVs to come on the deck. So, and that information is in our website under to the right of the screen, live data. You can see how deep Hercules is, and those numbers keep decreasing since we are ascending. So there was a viewer who asked that question. And Jason, thank you for all of your questions and comments. We appreciate your time with us. Yeah, there's a
So when you came up here before, kind of, you were on a sailboat, right? It was
cool.
This expedition you were on.
Yeah, we were on a I think it's called a 3-masted schooner sailboat. So yeah, definitely a way different experience. This is way more like luxurious.
Yeah, that's amazing.
Sure.
How long were you out for?
We were out here for a little over 2 weeks, so
what a
13
cool
days.
experience.
Or no, sorry, not even 2 weeks, like 13 days, almost 2 weeks. But yeah, it was amazing, especially for the first time for me to be able to come up here in that vessel in that way, and also learn how to sail. So yeah, that was really cool.
well, fun. And how deep in the ocean did you guys observe?
How deep was
How deep were you observing in the ocean?
Oh,
or was it just on
we
land?
actually did, yeah, we did shoreline and intertidal work. So we were just on the sailboat as our vessel to get out to our research area, I guess. Yeah.
And there was a contingent of Native Hawaiian researchers mostly, right? I mean, did did a whole really cool
Yeah.
ArcGIS story map about
Yeah.
that expedition.
Yeah, that was even more special because yeah, like you said, it was the first time that the research was led by a native Hawaiian crew, so very special experience. And yeah, I think Emil sent out a story map, so you can check it out and learn more about our cruise.
So Beth, we have Dr. Beth Orcutt here with us now. It's taking
Hello, everyone.
Chris
Good
is
morning.
taking a little break. So you were down there breaking up your rock samples yesterday. What was some observations that you came away with, and aside from a sore wrist from using the
It's not
mallet.
sore. It's just getting a workout. Yeah. So yesterday's dive was on an unnamed seamount, just on the southern border of the monument, and like the rocks we saw today, and we'll see.
I did see.
We got two different rocks that I subsampled in the lab. One of them, when we collected it on the seafloor, I was like, oh, that one's going to fall apart really easily. It looks like it's altered, nice thick crust on it. And then the second one that was collected, I was like, I'm not so sure. That one looks like it's going to be tough. It looks like it's a really strong rind. Well, I was totally wrong. Both the one that looked really strong was really weak. It crumbled very easily like a cookie. And the first one that I thought was going to be so easy was really a workout. Yeah, we're constantly keeps you guessing. So we'll see what the two rocks that were collected for our project to understand mineral alteration and mineral crust formation, how those look today.
How much of the rock are you, do you have to like crush up to get a good sample? Is it the whole thing or just a little amount?
Yeah, thanks for that question. Our project is to understand, really focus on the mineral crusts that form on the outside of the basalts on this seamount. And I need about 50 to 60 cubic centimeters for the various types of subsamples and analyses that we want to do. So one of the samples we collected on the first dive was about the size of my fist. And I needed most of that. I maybe took about a half to 2/3 of that material for the subsampling and reserved at least a third, if not more, of each of the samples for archival so that other researchers could potentially work on these samples as well.
Awesome.
Yeah.
That's cool. It has multiple purposes.
It does have multiple purposes. And hopefully when the analyses are done that I proposed, hopefully the residues could be used by somebody else as well. So that the samples really have a long history and reveal lots of knowledge about this environment.
That's awesome.
And how long does that process of analysis takes place? Is it
That's a
continuous?
sometimes that's a hard question to answer, because as new methods become available, we might want to try to apply them later. I'm not coming away from the ship with much data about the samples because most of the analyses have to happen in a laboratory setting with instruments that are not here. Yes. So let's imagine these samples come back in December to the laboratory after the expedition is over. It'll take us several weeks to process the samples for any individual analysis that we want to do, like measuring the amount of cells, measuring the amount of ATP, trying to extract DNA, sequence the DNA, analyze the DNA. So it'll take several months for us to generate kind of an initial picture of what the samples are telling us, and then we may then decide to dive deeper on a few analyses depending on what those initial analyses tell us. So generally, for instance, when we get funded to do this work, those grants are usually 2 to 3 years in length to allow the researchers enough time to get the samples, do the analyses, interpret the analyses, and communicate those results.
Yes, well, that's thank you for that answer.
Yeah.
Getting the big picture here.
Yeah.
When do you identify the types of organisms and numbers of organisms on a rock sample?
Yeah.
Does that come first and then?
We try to do the analyses in parallel. So we'll be not just doing one and then the next one, but kind of be moving forward many analyses. So there'll be several people in the lab helping with this work. For the microbial identification, what we'll do is take some of the rock sample and expose it to some chemicals to try to release the DNA molecules from all of the organic material and minerals that are in the rock sample. And then we'll take that DNA and purify it to get it nice and clean. And we will then, the first thing that we'll do is try to look for what's called a taxonomic marker, a particular gene sequence in that DNA that all microbial life has that gene. And we'll try to sequence that just that one gene, but from all the organisms that contributed DNA to that sample. And that will give us kind of an inventory of what the community looks like. How many of species A, is species B there, is species C there, and what's their relative abundance based on the amount of DNA.
I see. So the identification of these microbes is through DNA analysis?
It's definitely through DNA analysis. We'll also do some microscopy, but
yeah,
you can't really tell what a microbe is just by how it looks because their shapes are not very distinct. There's a few microbes that have a distinct shape, or they might precipitate a distinct mineral type. For instance, some of the organisms that are involved in the oxidation of iron and manganese, they precipitate rust, essentially, that has a particular shape. But that's very rare. Most microbes have no structural cues as to who they are.
Especially if we haven't seen them before. We don't know what they look like, right?
That's true. That's always that's always a challenge.
Oh, wow. Thank you. Thank you for
Yeah,
that
no trouble.
explanation. You also mentioned ATP.
Yes.
How do you identify the ATP amount, or how do you go about that? Do you count the number of?
Yeah, so that's a chemical analysis that we do. So ATP is kind of an energy molecule in all cells. And so we want to try to quantify it as a kind of indicator of how much life is in the sample. It's just one indicator. So for instance, cells that might be really active might have a lot of ATP, and samples that don't have a lot of activity, they're going to have a low amount of this molecule. And so the way that we do that analysis is, again, we take our sample and expose it to a liquid solution that has a lot of phosphate in it, so that it helps. ATP can be kind of a sticky molecule because it wants to bind to charged surfaces. And so we add phosphate to kind of buffer that stickiness. We'll freeze the samples, and then back at the laboratory, we will expose the sample to an extract or an enzyme that lights up in the presence of ATP. And this enzyme is actually derived from firefly, the enzymes that cause fireflies to light up. It's a very similar type of enzyme that's produced. So you have this enzyme. When it's in the presence of ATP, it luminesces, it glows. And so we put it in an instrument that measures light production. And that's how we get at a measure of ATP.
Wow, thank you for that explanation.
Yeah.
I was just showing how the molecule of ATP and ADP look like and which one has more energy. So
Okay.
Yeah, that's pretty fresh.
Yeah, some basic biology 101 there.
Yeah.
All right. Well, thanks for the questions. It was nice to pop into your shift and say hello, and also say hello to folks at home who are watching. But I need to go downstairs and get my sampling material ready so that once the ROV is on the boat, I can get those samples collected as quickly as possible.
And breakfast.
All right.
And breakfast.
Yes.
It is breakfast
Oh,
time.
yes.
Okay.
Well, thank you so much for that lovely presentation and
Yeah.
appearance.
No
Yeah.
problem.
Yeah, thank you.
I think so, yeah. He was going to bring back some coordinates for our next dive.
Okay, so we have like 20 more minutes for this dive to be over, and then 4 hours to unload the samples, get the ROVs ready, and then start the other shift, huh?
Yeah, around 12 o'clock.
It's going to be exciting. The next 2 shifts for us.
So what are you looking forward to teaching your students from this experience?
You know, I'm going to have to organize my curriculum now because I see like 2 big columns here of my lessons. I usually start with, well, this year I started showing my students the whale fall. Just what do you see? It's something that you haven't seen before. It catches their attention, and so ecology, but like for example, photosynthesis on land, and photosynthesis in the ocean. A food web on land and a food web in the ocean. And then now I'm going to add the microbes schematic that Beth shared with us, because I haven't seen that before. That's very colorful. We don't really think of microbes and supporting everybody on Earth, but there are all kinds of microbes. We have microbes in our
we have them from the stratosphere. They've found them as high up as all the way up in the Earth's atmosphere to the deepest parts of the ocean to our guts, and I find that part fascinating because they're finding what influence these microbes have on our entire emotional landscape. And,
yes, that's right.
Changes and how our gut microbe can trigger things like or be involved in things like depression and other
Where are they going?
emotional landscapes. So microbes are integral part of all life on Earth.
Everything was fine.
And yeah, I'm learning so much. So much more vocabulary, like biocultural is a new vocabulary for me.
All right, we're coming up on 300 meters.
Okay.
Bridge nav.
So yeah, and just the whole visitation to the model of the 4
it looks like we have
global
air
systems of Earth.
Captain on the bridge.
Atmosphere. We talk about oxygen.
Okay, and it looks like you've just made some heading changes.
The hydrosphere.
Happy with this heading?
The geosphere. I always just thought of the geosphere, the soils on like land, but there's geosphere throughout the ocean. So yeah, a whole new perspective to teaching. Right when I need it, so right in the middle of my career. So
it's great. And are your students following along on the expedition?
Some of them are following along. I got a couple of questions during my watch the other day, and now that I have more information of the science that is happening on our ship, I'm also sharing some pictures, and definitely some of them need extra credit, so yeah, so I'm adding to my folder of extra credit Thanksgiving assignments.
Nice.
But yeah, I can see their excitement,
they're very envious
about just what is your life like on the ship? Yeah.
Yeah, I'm sure there's a lot to talk about. Just all the different career pathways that are represented on this ship. Everything from the people who take care of the equipment, the engines, all the operations on the ship. You know, in a marine environment, everything decays so rapidly and so quickly.
We don't have to do the move.
That have to take care of that at all times.
If we get to it, great, but that's fine.
Lubricate all different aspects of the equipment, and painting, repainting the deck. There were people out repainting parts of the deck yesterday so it doesn't decay. And then we have chefs that are certainly preparing all the food for the people on board. The captain and the other crew that are responsible for driving the ship and for navigating, getting us safely from one place to another. And then we have our scientists on board that we've been focusing primarily on for this broadcast. And of course the ROV pilots, documentary crew. We have IT specialists on board. That there's a lot of computer equipment on board that needs regular attention. I don't even know. It'd be interesting to find out how many computers are actually on board this ship. I would say it's in the hundreds, if not near 1,000. I don't know. It's quite a bit of equipment.
Yes, and how? Well, just Hercules alone, I think, has 16 cameras.
Mm-hmm.
So, yes, it's a
Hercules has about 7 cameras, 7 or 8?
7 or 8 cameras.
Yeah.
Depending on the configuration.
Uh-huh.
1,
So
2, 3, 4, 5, 6, 7, 8 cameras.
8 cameras, wow. So a lot of ways to be involved in expeditions like this through many different careers.
Yeah, and our website, Nautilus Live, has a very useful structure. We can see the teams.
Mm-hmm.
That's where I go to show my students what kind of careers are available in ocean exploration. Well, just a few, I'm sure, but there is our whole team, and now I have a better idea of how UV Nautilus works too. We have 3 teams for 3 different shifts, two times, right, throughout the 24-hour. So we have 2 sets of scientists, 2 teams of scientists, 2 teams of navigators, 2 teams of data loggers, 2 teams of ROV pilots, 2 teams of video engineers, and we do our watch rotations.
We're finally starting to see some more life.
Looked like a squid.
Critters, yeah, that was definitely a squid. Little guy on the left-hand
Back
side.
trying to, down in my room trying to find some data, which I did not succeed at finding, so I'm going to have to do a little bit of searching around here.
Data mining.
I had a little issue with ARC yesterday, and this one file seems to be missing. And I did correct the problem, but I wonder if somehow that went away.
Okay, Roger.
But that's okay, but actually I realized that I need to do some adjustments anyway to this. These are prospective dive sites. But anyway, you know, one thing I couldn't find also, which I'm very disappointed, I purposely brought out my Papahanaumokuakea Marine National Monument notepad to write stuff down and notes for the, and I know I had it, and I can't find that either. So I think
so we're going to stop tracking a line at 10 meters, is that right?
I think there's a little hole in the room, and things are finding their way into this little hole. Which is not surprising.
Roger.
You know?
Okay. No. Sounds great.
Anyway, I am now thinking hard as to where I put my pad and where my data file is. It's somewhere there.
Here's my data.
My data.
Looks like there's going to be a bit of current pushing me that way. So we'll see how that goes. which is the right way to be pushed. See if I can get a goal.
So the sun is coming up outside, and we're about 179 meters from the surface.
Totally. Yeah, absolutely. That was also a lateral move, so it's not exclusively current, but it seemed to move faster than expected. So we'll see how it goes. Plus, 170 meters is entirely different than the surface, so who knows what's going to happen.
I think maybe we can go ahead and let it go. They're going to need to be talking to each other pretty soon,
Okay.
and I'm wondering whether we should probably head on down and get some breakfast.
All right. Well, signing off until our next dive.
Yeah, we're going to sign off. We're about to get the ROV on the surface, and we're going to let the ROV team do their thing. So thank all of you for joining us, and we're going to see you shortly.
Thanks for all the fantastic stories for the blue water.
Yeah,
Yes,
thank you,
thank you,
Trevor. Thank you for a
everybody.
great dive.
All right, 160 meters.
Cool. That's pretty, 9 minutes to surface. That's pretty bang on.
I mean, what can I say?
Good calculator.
Thanks.
That was very good.
Good driving. Gotta keep Mark happy.
Yeah. Okay, how can I make this go a little better? Probably like that. What is happening? Why is this one? Yeah. Sure. Just enough to get that thumb cramp. Yeah. See you in the gym just lifting thumb weights. Like, what? What is she doing? Yeah, so I'm lateraling to my port,
Roger.
and it looks like we're going pretty straight, which is fine. Just another, anyway, it's fine.
If it becomes unfine,
We'll change something.
we'll change something.
Yeah.
Standing by.
Okay, I'm going to do the recover salvo. Keep an eye on things.
Now I'm curious.
You can already hear
Oh,
it.
that one, yeah. Terrible.
Yeah, not my favorite.
It's a bit swelly out there, I guess. Big swells. I quite like that view.
Okay. Yeah, I think we're going to come up to starboard. Sorry, my starboard.
Your starboard.
Shipboard.
I'm like, they're still tweaking it. I'm not really sure why.
Okay, now I'm making some headway.
Antonella, can you try your latch on radio? Okay, cool. Yep.
This has been getting like reasonable soundings to 10 meters.
Yeah, let's keep it going then.
It's amazing.
That's awesome.
So nice.
And kill it once we're on the surface. This is the number they'll stop at, not this one. They're different.
That is?
That's the one, that's the winch payout.
Okay, you ready? You okay to come up? Control, yeah, we're okay to come up. I hate when he beats me. We'll work on it.
Yeah, that's the trick, is watch this one.
Yeah.
But also know that it's a little bit delayed.
We'll get him next time.
Call us back, so they're not coming up right now.
Deck bridge. Deck, go ahead. Is ready. Start coming up.
Who's on deck? Jake and who?
Yeah, who is that?
Is it Kate?
Oh, it is Kate. Yeah, good
Seems
job,
like
Kate.
not the time that Kate would be awake. Isn't she 12 to 4?
She is 12 to 4.
Perfect.
Yeah, well, she goes to the gym, she takes a nap, so, and she wanted to get out on deck, so cool, good for her.
Okay, wow. They're coming out 12, 13, 14. Oh. Randy's in a big hurry.
This is going to be probably her first hooking experience. We're judging you, Kate.
She's a lefty.
I wonder, actually, I don't know.
I'm a lefty for that, like hockey.
That's how I stand on that side, though.
Are you a lefty for hockey?
No?
Oh.
I think that's how I stand. I don't know.
Argus is beating me. Rennie's giving her 16 meters a minute. I say Rennie, I don't actually know that it's Rennie. It's gotta be Rennie.
Oh, and winch, yeah, it has to be, because he was wandering around and now.
Yeah, he was wandering around and nowhere to be seen. Not like him. He wanted to set a recovery speed record. Normally I'd ask him to slow down, but I'm hungry, so it's fine.
Right.
There's the ship. Alas.
Bridge nav. Can we reduce power on the jump pump? Thank you. You want me to stop the meso?
Yeah, sure, please. Does the stop button do both heads?
Oh, I don't know.
Is that what you pushed?
Yeah.
Cool.
Did it work?
It seemed to work.
Seemed to work.
I didn't know that. Oh, so fast. Yeah, gonna get a goal. I think.
Oh, for sure.
Yeah. I'm going to my starboard though, for sure. Okay. See the hooking. Come on, yeah, there you go. That's the better technique. Nice.
Nice, well done.
Can you please put bubble on the res? And I can't see bubble anyway. Huh, perfect. There we go. Oh yeah, that's right. Okay, good.
Alright, killed the beacons.
Roger.
Bridge nav. Can we bring jet pump back up to 90% and hold position? Thank you.
Nice job, Antonella. You got all the things. When I turned off bypass early, so I tricked you, did the opposite thing than what I said, and the voltages look good. I turned it off a lot earlier than I said because the pressure is getting high. I don't think so. I think that'll be all right. So you can write those voltages down though. Yeah, 124, 132, 132. Then the next watches can know what's going on too. Team ratchet strap. Here we go.
I cannot ratchet strap under observation. Like I screw it up every single time. If
Do you
somebody's
really?
oh yeah, if someone's watching me, like every time they're
You
gonna
know
be like,
like the easy trick? You just have it closed, fully closed. You don't have it open.
Yeah?
Otherwise, you had to weave it back.
Oh,
through.
and then slide it through. Yeah,
Just
yeah.
go.
You know, that's exactly what Josh shows me every time, and I still screw it up every single time.
Fair enough. Yeah. And some people start with it open and then weave it back through, which is fine, but it's an extra step. And
Yeah, yeah.
Someone's got a power stance.
Hmm?
So there's quite a power stance right there.
Yeah.
Oh, we got one on deck by 8, kind of.
Got one what?
We got at least one vehicle on deck by 8. We were close.
Well, recovery started at 8, I think.
Oh,
That,
does that
that's a
count?
whole, to say, you know, recovery on it at 8 is a pretty broad thing.
I guess the only thing that matters is, did Mark have breakfast?
Yeah.
that's the true measure of success, and how the rest of the day is going to go.
Right. I also hope to
Have breakfast.
have breakfast.
Yeah.
Receive said meal. That's good to do, yeah. It's not a must, but it's a good to do. Beauty, yeah. Optimize. Nice. I don't need no stinking lights. I don't need no stinking lights. What's that? I can't hear you at all.
A version of Ubuntu?
Oh.
I don't know. Where would one look? I know how to do like 3 things on this computer.
Those are not terminals, those are shell blah blah blahs. That's what's making this go right now. Oh no, that's what's making that go right now. I don't know.
And then what? LSB. Tab, thank you. And then
Why
what?
tab? Yeah.
Okay.
Herc, you want to drive to the end of your tether?
Sure.
Roger. What? Too hard. Next blue water, we can investigate this one.
Come on, football. There we go.
Okay, Herc, keep a little tension on it.
I'm giving her all she's got.
Roger.
I'm trying to do it in bypass, which is not much tension. Keep bumping it, I guess.